Okay, good morning, students. I hope I'm audible to you all. And I guess 14 students are in the meeting and 13 students, 14 including me. A few students are yet to join, right? So, in the meanwhile, we can just discuss briefly the last practical that we did. So, in the last practical, we discussed about the mechanical transmission. So, who is going to tell me that what is mechanical transmission of viruses? Yes, Manaswani.
Ma'am, so mechanical transmission is a way of movement of the virus from one host to another through either
Mhm.
some cut in the plant or some kind of rubbing, but there's a
Mhm.
physical contact, and
Yeah.
the virus is transmitted in that manner.
Okay. Fine. Okay, Manaswani. So, yes, my next question is basically that what kind of a buffers are or what kind of a buffer is recommended for mechanical transmission when we prepare the extract, right? We prepare the extract or inoculum from the infected plant. So, what kind of a buffer which is being recommended? Yes, no one? Phosphate buffers at particular pH was mentioned that we need this kind of a buffer for the extraction of the plant and for the inoculum preparation when we do the homogenization or a grinding of the tissue in a mortar vessel. So particular buffer is required, right? So it's a potassium phosphate buffer or a phosphate buffer, right? And okay, the next question is that why we add PVP, polyvinyl pyrrolidine, to the extraction buffer? What is its use? What is its role in the plant extraction buffer? No one?
Ma'am, can I
Yes.
answer?
Yes, Yukta, please go on.
Um, yes, ma'am. So polyvinyl PVP is added. It acts as a reducing agent, so it is added
Mhm.
to the buffer to protect the virus against phenols if they are present in the host.
Yes, you're right, Yukta, that we add the PVP to the extraction buffer to save a virus against the different phenols which are being present in the different plants. Maybe some kind of sometimes tannins are present in the plant. So, in order to so that they don't hinder in the extraction of our virus, right? So we add that, right? So okay, the next question is that how we provide wound to the plant for the virus transmission? What kind of substance is being used or a chemical is being used? How do we do that? Because we need to provide some kind of a wound to the plant so that virus can enter. So how do we do that?
Ma'am, for wounds we use abrasive powders like carborundum or cerebrite.
Yes, right. For the wounds, we use certain kind of abrasive powders such as carborundum powder. They are very fine powders and very in a minute quantity. We need to use those kind of a powder, right? Okay, fine. So we discussed about the mechanical transmission. Mechanical transmission is at the earlier time it was one of the important way to study the biological properties of the virus also when the molecular methods were not identified. And right now also if we want to maintain the virus on the host plant for other molecular studies, we need to maintain them on some propagative host or the other host plants such as Nicotiana tabacum or other species which can be grown in the polyhouses, and they can be maintained on that so that as and when required we can have the pure viruses with us, right? So that can be done. The host plants are Chinopodium faeciolus or Nicotiana tabacum or Nicotiana xanthomana, which are being very commonly used. So we discussed the protocol also, and I've given you the experiment to explain the mechanical transmission of CMV from cucurbitaceae host to Nicotiana tabacum plant. So I'm sure you must have, I don't know, in the morning I was checking, I created one special assignment to submit your practical notebook. So after this first class is over, all of you please submit your practical notebooks there, and those who have not done or completed their practical files, please just do this in today's second half of the practical. So you complete those file, and so at the end, those students will get attendance only those who have submitted their both practicals. So make one PDF and submit, and they will get attendance for the second class. Only those students, those who have submitted their files properly done, right? So okay, to start with today's next experiment, it is very interesting one, and it was very commonly done when students, when we first start working on the viruses or a plant viruses in general. So first thing we usually do is a symptomatology, second thing we do the mechanical transmission, and then third thing in we are trying to go in a sequence manner, that okay, now virus is being transmitted, now you want to check whether the virus has been transmitted or not. So for what we need, what how we will proceed further, what can be our next step, any guesses, any thought on this, that what would be our next step for the identification of the virus, what we'll do next? Are you all sleeping? Please, if so, then good morning and wake up. This is just your third class, I guess, since morning. So you don't know, I mean, the sample is there and a virus has been inoculated. So what next we'll do to identify that it is the same virus that we have inoculated? क्योंकि chances हैं कि वो virus नहीं होगा, कुछ और होगा, तो हम क्या करेंगे अब?
Ma'am, we
Yes,
can test it by
yes,
ELISA or
Ayush. One by one. Yes, Ayushi, go on.
Ma'am, we can test it by ELISA or PCR technique.
Yes. Okay, fine. Right. We'll test it by ELISA or a PCR. Okay, I have selected a PCR. I want to go for a PCR. Now what will I do? What will be my first step? Yes, Yukta.
DNA isolation from the.
Pardon? I can't hear you, Yukta.
Ma'am, we'll
Yes,
first
anyone?
isolate the DNA.
Yes, right. So depending upon the genome of the virus, whether it is a RNA virus or a DNA virus, we will isolate RNA or a DNA from the plant tissue. So this is the next step that we are going to do today, that we are isolating. We will study the protocol for the isolation of the DNA and RNA. There are different protocols. Certain chemicals are differently used. Otherwise, isolation can be done by the similar methods also, RNA and DNA. So at the end, we can add certain enzymes to inhibit the RNA if we want a DNA, or if we want a RNA, we can add DNases at the end, right? So to isolate RNA or a DNA from a plant, there are a lot of methods available, right? There are classical methods. Classical methods are basically referred to the methods in which we use certain kind of a reagents, and certain kind of a reagents and certain kind of a chemicals are being used, and this process is a bit lengthy as compared to the kit method. So usually the normal protocol that we follow for the teaching purposes or the DNA isolation is the classical CTAB method. CTAB is a chemical I'll be discussing later, that what is its function. So we use that method, right? Usually this is a lengthy method, but yes, this protocol gives a good quality of a DNA. But then sometimes for the research purposes, we need a purified form of a DNA or a RNA or a nucleic acid. So yes, the nucleic acids which are extracted by using classical methods are usually sometimes not in a very purified form, right? If you want to do certain kind of a higher experiments or a molecular study. So sometimes we go for, if we have a fundings available, we have a fund, so we can go for the kit method. So kits are basically, they are being standardized and they are being developed by the certain companies, or rather many companies. So they, in the kit, a different kind of a buffer, maybe 1, 2, 3 form, they'll just name it like that. They'll not tell us what are the components in the buffer. And it is basically there are columns, silica columns, which are made up of some silica or some other material which can bind to DNA or RNA. So in this way, there are separate methods or a procedure is being followed in which our sample is passed through those columns. Those columns they bind the DNA or RNA, and then they are being washed through using several buffers. And then at the end, we use a certain kind of a elutant which can elute the DNA or RNA from the column at the end. So these are the simpler method, more accurate method to get very purified form of a DNA. But then definitely a lot of funding is being required because they cost a lot. Gel based method or a column based method, they cost a lot. So usually when we do a normal routine work or uh, research work or a teaching work, just a moment. So usually what happens that we we just a moment, I'm getting call from other meeting. I need to tell them. Just give me 2 minutes. Okay, sorry for that. There was another call to join another meeting. So, okay, so we were discussing that, yes, there are different methods. There are kit-based method, there are classical methods. So basically, when we are teaching students how to extract DNA or RNA for the first time, we prefer the classical methods by using different chemicals, right? So those methods are being followed or those methods are being used. But then later, if we want to use those DNA and RNA for the research purposes, then we go for the kit-based methods also, right? So isolating DNA from a plant tissue is a different, yes, different plant, they have a different component. Some plants, they have a more of secondary metabolites such as phenolics or tannins, whereas other plants, they don't have. So there are separate methods which are being standardized for such kind of a plant by addition of certain kind of a chemicals, and those chemicals might not be required when you're doing for a normal plant where the not much of phenolics or tannins or a secondary metabolites are not present, right? So unlike animal tissue, what happens, the same tissue type from different species, they have the same characteristics, right? Animal tissue, every kind of animal tissue is being processed and treated in a similar way. Plants, they have a variable level of metabolites and biomolecules. Polysaccharides and polyphenols are basically two classes of a plant biomolecules that vary widely between the species, and they cause a lot of problem when isolating DNA. So contaminating polysaccharides and polyphenols, they can interfere with manipulation of DNA following its isolation. So we need a purified form of a DNA. We don't want to use DNA which is contaminated with these kind of substances for our molecular or further studies. So majority, there are two factors which are responsible for difficulties in plant DNA extraction: the difference in structure of the plant cell, the component of the plant cell. These are very different from the animal cell, right? The structure of the plant cell is different than animal cell. We all know the plant cells, they have a rigid cell wall, whereas animal cells, they do not have any cell wall. So animal cells, they have a very smooth cell membrane, and plant cells, they have a hard, rough, and a solid cell wall. So here, the initial starting at at the onset of the practical, basically, we need to break this cell, right, in order to release the components of the cell along with our DNA or RNA. So there are different methods which are available and which can remove these polysaccharides and polyphenols from different DNA preparations. CTAB. CTAB is a cetyl trimethyl ammonium bromide. This is one most important CTAB buffer, where CTAB is the main ingredient. This detergent, it helps in the separation of polysaccharides during purifications, while certain other additive compounds such as polyvinyl pyrrolidine, they can aid in removing of the polyphenols. So CTAB extraction based buffers are very commonly used when purifying DNA from the plant tissue. So one option for purifying DNA using CTAB exploits that polysaccharides, they have a different kind of a solubilities in a CTAB depending upon the concentration of the sodium chloride which is being used. So at what happens at higher salt concentration, the polysaccharides are insoluble, while at lower salt concentration, the DNA is insoluble. So by varying these conditions, we know which one we want. We can use the different concentration and we can precipitate out our DNA or RNA. So consequently, by adjusting salt concentration in the lysates which contain CTAB, polysaccharides and DNA can be precipitated in a separate way. They both can be precipitated by using different kind of chemicals. So polyphenols, these contain more than, these are the compound, they have a phenolic ring and they bind very efficiently to the DNA. So they are occurring naturally in the plants, but then sometimes they are being also produced in the plants due to the tissue damage, browning kind of a thing is caused. So these are all polyphenols. Sometimes we keep the our apples or some fruits cut for a longer time. The browning happens in them also. So that is also due to the phenolics basically, right? So what happened upon homogenization of plant tissues, these polyphenols are synthesized by liberated polyphenol oxidase. So when we add PVP, polyvinyl pyrrolidine, it prevents the interaction of DNA and polyphenolic ring by binding up to the polyphenol. So polyphenols cannot be bind to the DNA and these polyphenols can be precipitated. So we know the plant cell wall there's a lot of components. There's a chitin, there's a cellulose, right? So these kind of a components, they give extra strength to the cell wall. It is more rigid and due to the rigid nature of the plant cell wall, the shape of the plant cell is very fixed. So whereas the cell membrane of the animal cell is very smooth. So hence we don't require much to do the breaking the cell membrane, not much strength or not much enzymes or not much other methods that we use in case plant is required in this case. So yes, this also we know that the components of plant cells are different than the animal cell. And then plant cells, they have a chloroplast. So chloroplast is involved in the photosynthesis. So then photosynthesis takes place, the secondary metabolites are also produced, and these secondary metabolites are absent in the animal cell. So this is just a simple method which says that extracting DNA from plant cell, we need to disrupt the cell wall. We need to remove or break the cell wall or a cell membrane with the help of certain chemical or a physical methods. All this process together is called a DNA extraction. So what are the different steps and what are the different chemicals and what are the different buffers that we use in the DNA extraction? So lysis of the cell wall can be done by using certain chemicals or physical methods. Physical method, what is the physical method that we commonly use? We grind them. So we take a pestle and mortar, we use a certain kind of a liquid nitrogen, because when we use a liquid nitrogen, they freeze the plant tissue all at once. So the grinding them to a powdered form is very easily when we do this, right? So then we need to separate DNA from other organelles. That can be done by CTAB and other chemicals. Then at the end, we need to precipitate DNA out of it, right? Once it is being separated, now we want to take it out. So that is again done by using alcohol or isopropanol, a different kind of chemicals. Then purification of extracted DNA by 70% alcohol at the end. And we can dissolve the DNA in a water also, or we can dissolve the DNA in a tris or a EDTA buffer that we prepare. So when we dissolve them in a EDTA buffer or some kind of a buffer, they don't degrade very easily. I mean, we can use them for a longer time. Whereas if we dissolve them in some kind of a water, or water is also sterile water, or some iron-free waters which are available. So in that also we can, but then their life decreases. They can be degraded after some time. So CTAB, acetyl trimethyl ammonium bromide, also called cetrimonium bromide. This is a cationic detergent and it is very commonly used in DNA extraction. This agent is soluble both in water and alcohol also, and this is very corrosive and toxic in nature. So when we prepare buffers out of these kind of reagents, or whether, see, whenever we enter a lab, I'll be discussing the lab biosafety with you in the third module. So whenever we enter the lab, it is very important that we wear gloves, we wear lab coat, and we don't touch anything by the bare hands, because the chemicals can be corrosive, they can be carcinogenic, they can be lot of problems that we can have if we touch these chemicals by hand, right? So the cell wall contains mainly plant cell walls, they have a hydrophilic end group. So detergents such as CTAP, they can dissolve both polar as well as non-polar compounds also. They can bind with the cell wall polysaccharides and protein, and by complexing with it, it co-precipitates the macromolecules along with the DNA. So it is precipitating along with the other macromolecules or other components of the plants also, then which can be later removed by the different processes of the centrifugation and by adding certain chemicals which can precipitate DNA at the end, right? So there are certain solvents that we use which if we need DNA, so they'll precipitate proteins separately. We'll get a DNA in an aqueous phase. Proteins are being in the lower phase. So we can just take out our DNA from the aqueous phase, and again the several washing steps are being done to precipitate DNA or to purify it further. So role of liquid nitrogen in plant DNA extraction, as I told you that it is recommended. Usually what happens, either we go and get fresh samples, right? Fresh samples, we get it in the ice and we just go directly crushing. Sometimes what happens that samples are being stored in the -80 or -20 for a longer time. So they are already in a freezed form. So we don't want them to thaw, because thawing can result in the loss of the viruses or a DNA, degradation of a RNA or a DNA. Though DNA degradation does not take place that easily, but RNA is degraded very commonly. So we need to maintain that temperature, right? So liquid nitrogen, during grinding, the heat is generated, which activates the enzyme such as endonucleases or exonucleases, and these enzymes can cleave our DNA into smaller pieces. So what happens when we add liquid nitrogen, this prevents the enzymatic reaction also. Also the liquid nitrogen, it frees the tissue. So by once the tissue are being frozen, to make it into a fine powder of that tissue becomes much easier, and it helps in making homogeneous mixture of the tissue. It instantly froze the tissue to the point where it becomes ash, like it can be just come into a powdered form. Then tannin, the villain of plant DNA extraction. This is again a long chain of phenolic ring, which are the major constituents of polyphenolic compound. And this is naturally occurring in the plant, but then when tissue damage is there, in those conditions, these are secreted more. So they again, these kind of polyphenolic compounds, they inhibit the PCR or polymerase chain reactions also. So they are not performed very accurately. So PVP binds to the phenolic ring of the tannin, and it prevents its interaction with DNA and protects it. So usually we add a PVP also to the buffer, so that it can prevent the losses which are caused by the polyphenolics or tannins. Hence, PVP is the main ingredient along with CTAP for the plant, which contains high level of tannin or a polyphenolic compounds. Sometimes beta-mercaptoethanol can also be playing the same role. So this chemical also plays the same role. So it is used in antioxidant, and again it prevents the oxidation of polyphenolic compound, and it helps in removing or separating it. So we can use this particular chemical also. So in general, 2% CTAP solution is used for plant DNA extraction. So there's a 2% CTAP, is a 0.7 molar NaCl and double distilled water, which is being final constituent can be made according to our requirement of the buffer. So CTAP extraction buffer, they have a 2% CTAP, 100 millimolar trace, EDTA, NaCl, or if required, PVP. This is optional. Then beta-mercaptoethanol is also optional if required. Then these are, I'm not going to discuss them in detail. These are how do we prepare 1 molar trace at pH 8.0. So I'm sure you all know how to prepare 1 molar solution of any compound or any substance, right? So this is, usually we prepare stocks. When we prepare trace or EDTA, we prepare a stock solution, and from that we use it for the making buffer. So CTAP method is a very simple. And at the end, we can add RNAs also, since we are extracting DNA. So through these methods, the nucleic acids are being extracted. So if we are interested in DNA, so we can add RNAs, so that will inhibit any kind of RNA contamination. So usually the procedure is that we take a quantified amount of a plant tissue, right? Here they have, the procedure is different. If we take 100-gram larger sample RNA preparation, then the procedure quantity of the buffers will increase. So usually we take a very smaller amount, 5 gram of a tissue. Will be cut into small pieces. We can add a liquid nitrogen to the tissue and we'll grind it roughly in the mortar and pestle. And these mortar and pestles are pre-sterilized in a hot air oven at a certain temperature, or sometimes we do it by autoclaving also. So after the tissue it becomes a powder, we add a 500 microliter of a CTAB extraction buffer, and we can add beta methylphenol if it is required, and then we grind it for some time. And vortexing is done by a small equipment, tabletop equipment, which mixes the components. So we can put it in a vortex, it will shake and it will mix the components of the buffer. So after it becomes homogeneous, we take the mixture into a new tube. Then we'll boil the sample or incubate the sample at 30 minutes at 60 degrees Celsius to 65 degrees Celsius. So after that, we'll centrifuge again at a certain RPM, maybe 25,000 or 20,000 for 5 to 8 minutes, and transfer the supernatant to the other tube. So debris, they get collected at the bottom, and we just need the supernatant, the upper part, which is more clearer, right? It's clearer and the transparent one. So this here a lot of we need to be very take a lot of precautions, because we are using a micropipettes. So if tips they go and touch the bottom debris, so that will contaminate our DNA or a nucleic acid or RNA that we are taking out. So we need to be very careful. At the end, we can leave certain amount of the supernatant also. So it's not that we need to extract all. Leave the little lower amount of the supernatant so that we don't get any debris contamination, right? So we need to be careful. We need only supernatant. We don't want debris in this. Then we can add some RNA solution to the supernatant. Again, incubate it for 37 degrees to 25 minutes. RNA is being added sometimes to remove the DNA and other component. Then centrifuge the samples at 10,000 RPM for 2 minutes, and then we take the supernatant to other tubes. And one thing I just want to add here, that CTAB method is being standardized for a different plant. There is one common method which is being published by the workers, the authors, and that method is being standardized by different people by adding, by modifying the the centrifugation step. Maybe some are doing it for a 10-minute lower time. So it is being modified according to the plant tissue that they are already working, right? So at the end, we add isopropanol to the supernatant, and we precipitate the DNA. We mix it. We try to mix it for a several times. Then again we centrifuge and we collect the pellet. This pellet is not visible usually because DNA we cannot see, right? So it is just transparent usually. Sometimes due to the contamination still present or purification is not very clear. The clarification steps are not done properly, so we can see the green pellet, right? So then again, we need to wash the DNA with the alcohol, or 80 to 90% alcohol. And then finally, we get the pellet will appear and we can dissolve it in the tea, buffer or elution. And then we can dissolve the DNA in it, right? So this is the one standardized protocol, or one common protocol, or a basic protocol. Now this protocol, when you see the lot of papers or a different kind of workers, they have modified these protocols according to their own needs. So it is not that one standard protocol. Yes, this is a standard for any plants or normal plant, a DNA can be extracted, but people have standardized this method also to get a more purified of a DNA according to their need, or according to the plant they are already working, right? So this is just a diagrammatic view which says that you take a tissue, you grind it in a mortar and a pestle, then you collect the homogeneous mixture in a particular tube, you centrifuge it, you collect the supernatant, then you add ethanol, right? So one particular step, sometimes isopropanol and chloroform is also being added. In some cases, they add this chemical. Again, this particular chemical, it separates the DNA from the protein or other mixtures, right? So it separates the DNA out in a separate aqueous phase. So this sometimes this is also being used. So how do we check the purity of the extracted DNA? There are several methods. The easiest one that we usually go for is we run the sample on the Agarose 1% Agarose gel, and we just see the band. So these the single kind of a high molecular weight bands we see at the top, right? So this shows that yes, the DNA at the lower side, in some cases you see some kind of a hazy thing, so that shows the some kind of impurities, maybe it's not properly done. So by this we can judge that okay, in the lane 5, 6, 7, 8 they are fine, but earlier lanes they don't seem, lane 3 seems to have a 2 band. So these are usually it could be in lane 3, if we are seeing 2 bands or more than that, it could be a RNA contamination also, right? So we check on the gel, and on that we see whether we can use this for the further processes or not. That can be done. Then another method, I'm not discussing, this is the method which is being given at the molecular manufacturer's instruction. So if I have got a kit from there are Kaizen, or there are other companies, RBC, there are a lot of companies which sell kits, right? So the buffers will come, there are columns in the kit. So you will just use the protocol. They have given a standardized protocol, and we'll just use. So advantages of using DNA isolation kits over crude method, that they are fast. Within half an hour, I guess we can get. Whereas if we go for a CTAP, sometimes it takes more than 1 day or so. So it's a longer procedure, right? And they do not contain harmful chemicals such as phenol, chloroform, precipitations. We don't need to involve. So minimum stepwise procedure, follow the instruction, and at the end, DNA or RNA is given. So this uses spin columns which contain silica gel-based membrane, and the silica basically binds to the nucleic acid. So by using separate different steps of a washings are being done to further make our DNA and RNA, purify or clean it from the debris. So there are different steps which are being done in this. So this these kind of methods are based on the columns. They use columns, fine columns. So these are costly also as compared to the CT method, right? So here the DNA can bound to the membrane, then it can be what there are usually 2 columns are being provided sometimes in this kind of a kit. So first, in the first steps, similar step is there. You crush the sample in a buffer, you pass it through the column, right? So we take the flow through. Now that flow through is again passed through the several buffers to clarify it further. Finally, again we take another column which will bind the DNA. So again that purified samples or a supernatant that we get from or flow through basically in the column, that is passed through the another column which will bind the DNA. Now it will bind the DNA. DNA is bound to those silica gels. Then different elution methods are being used to elute DNA from those gels basically, or the columns. And then further they provide the elution buffers also. You elute it and you use it for the further. So there are simpler protocols that we give. I'm not going to discuss this, and neither you need to write this protocol. This is just for the reference that what we do in a classical method and what we do in the commercial kits which are available. So everything is being provided by them. We just need to follow the protocol in this case, nothing else, right? So again, there are we can check the quality and quantity of DNA either by the gel method or by the absorbance quantification method, right? So if we have a spectrophotometer, we can take the OD of the sample or the DNA which is being extracted, and by different calculations we can see whether our DNA is in a purified form or there are some kind of a contamination which is present. So nucleic acid quantification is performed. There's a spectrophotometer, there's a qubit spectrophotometer, right? Then at basically 260 nanometer is the absorbance peaks for DNA and RNA we use. So at 260 nanometer we check their OD, right? So these are being commonly used method where spectrophotometer is available, right? So both DNA and RNA, they absorb light at 260 nanometer. So this measures the total nucleic acid. So nucleic acid samples are typically measured at 280 nanometer also, which gives the protein basically, right? So usually हम ratio लेते हैं 260 by 280 nanometer. So this will give us the value also if there's any protein contamination, because we are isolating the nucleic acid, and there are chances that proteins are not precipitated properly and they are coming along with the nucleic acid that we have extracted. So that their ratio will tell us about the protein contamination also. So along with the calculation of nucleic acid concentration, it is also very important to determine the purity of the sample for any kind of application. So proteins and other contaminants such as phenol and salts which are associated with the extraction process, they can interfere with the result, right? So again, the purity of this nucleic acid sample is determined by finding the ratio of nucleic acid measurement, which is being done at 260 nanometer, and protein measurement at 280 nanometer, right? So A to 60 by 280 ratio is a approximation of the purity that will approach the ideal ratios. For example, here, so pure DNA will show around somewhere 1.8 ratio, pure RNA around 2.0, and pure protein around 0.57. So by checking the values for our sample, we can interpret whether our DNA is a pure, whether there is a RNA contamination, or whether there is a protein contamination, right? So usually before we proceed for any kind of a molecular reaction or for certain processes, we need a certain quantified amount of a DNA, right? That 2 nanogram or a 3 nanogram or maybe more than that. For those kind of calculations, we need to quantify the DNA, right? It is very important. So DNA isolation is very simple procedure, and another guidelines or other some do's and don'ts are like when we work with the DNA and RNA. Usually in a high-end research lab, there are cold rooms, right? DNA is not degraded that fast, but RNA, they get degraded very easily. So either at all the time, we try to keep these columns and these ependophs in the ice or on the ice so that the temperature is being maintained. Or usually in some labs, there are cold rooms are available. So in the cold rooms, you go for a DNA or RNA section. So that will maintain the integrity of the DNA or RNA, and they will not be any degraded form. And another important thing is like to check them, we go for Agarose Gel Electrophoresis. So that will do the next experiment. So Agarose Gel Electrophoresis again involves the use of a carcinogenic chemicals such as ethidium bromide to stain the gel. These days, some other kind of a fluorescence methods or things are available, but then they are costly. ETBR is the cheapest solution available, which has been used since ages. It's just that we need to be careful while we handle with the ETBR. It is a carcinogenic. It can bind with our DNA and it can cause some cancer like things also. So it is not very good. So we always say that when you handle such kind of a gel, you should be very careful. So we need to be careful that how we discard these things. We need to be careful that when we prepare gel, we are wearing a gloves. When we are checking the gel in the Digidogs, there are Digidogs instrument equipment which comes there. Sometimes UV transilluminators also. So we put our gel which is stained with the ETBR in that, and it will fluoresce and give orange color bands if some kind of amplification is there, right? So this is how we check this. So precautions are wear gloves, wear lab coats, be careful while you are grinding, be careful while you are separating and taking the two different phases are being there and you need one phase, or when we are taking the supernatant in order to avoid the contamination, right? So also we need to be careful when we prepare buffers. Buffers should be properly autoclaved, especially the parts which can be autoclaved, right? So we usually prepare stocks of the different ingredients and then we mix them. And if autoclaving is required further earlier, then we do it earlier, or if we can do in a full form of a buffer, it can be done. So autoclaved, sterilized buffer, sterilized cassette and mortars, proper glassware, proper gloves, all these are always a thumb rule for any kind of a molecular biology experiment or a DNA and RNA extraction that you do in the research lab, right? So this was about the DNA, RNA extraction. RNA extraction protocols are also similar. They are not much different. Sometimes these reagents are different. Triazole reagent that we use sometimes for the RNA extraction, or there are the columns also which are available for the RNA extraction, right? So usually this is a stepwise procedure that you isolate the DNA. Now next step would be, okay, Agarose Gel Electrophoresis, we have checked the quantity and quality of our DNA. And the next would be the PCR or RT-PCR. PCR if DNA is our virus, plant virus, DNA virus, or if it is a RNA virus, then reverse transcript, RT-PCR, reverse transcription polymerase chain reaction. So before PCR, we do prepare a complementary DNA out of the RNA, and then we go for the PCR, right? So one step is more in case of RNA viruses. So for that also, we have a DNA, but then we need primers also. So have you studied in any of your previous lectures that how do we prepare oligonucleotides or primers for identification of any genes? Anyone? Have you studied in some practicals, molecular biology or bioinformatics or anywhere? Yes, Manasani? Yes or not? Because if you have not studied, then I will tell you how we prepare primers and how we make design primers basically.
Ma'am,
I'll
so
I'll
see, the thing we didn't learn it in like practically, but we know like what primers are and how they're
okay.
used, but we don't know about design.
Okay, you don't know. Okay, then no problem. I'll take one class. It is very interesting. It is all being done by using bioinformatic tools. So one lecture I can take on that. Primer designing is very important, and I think interesting part also that you should know how to design a specific primer and then how to design a degenerate primer, right? So specific primers, they are specifically for one particular gene. You see the sequence, forward sequence and the reverse sequence of a gene, and then 15 to 20 base pair, you make a primers, two primers. But degenerate primers are the primers which can identify viruses in a within a one genera, right? There's a big virus, for example. So they have a certain conserved regions amongst themselves. So we design primers for those conserved regions, and those primers are able to identify, maybe primers are designed for the core protein gene or other genes. So I'll take one practical on this. So where I'll tell you that how what kind of a sequences we select when we design primers, what kind of a softwares are available online, free also they are available. There's a primer free, or there are other software will put our primer, and what are the different precautions that we need to take. I mean, when we are preparing primer, what should be the melting point, what should be the annealing temperature. So all those things are being taken into account. What should be the Gs to C ratio in the primer? How we cannot increase Gs to C in a primer because that will increase the annealing temperature. Increase in annealing temperature will further the targeted primers, they will not bind properly if we are increasing the annealing temperature. At a higher temperature, they will not bind, and at lower temperature, annealing temperature, if it's there, then anything can bind at that temperature. So this is why a specific annealing temperature is very important for a primers. We need to prepare primers in such a way that there should be a particular average annealing temperature so that they can bind very easily and in a proper way to our targeting. So I'll take one class for that. So we will see if we can do that primer designing. So I'll tell you in one class, I'll tell you that how we do it. And in next class, I'll give you different viruses, maybe gene, I'll tell you, and then you'll prepare primers for that own in your own way, the technique that I'll tell you, and then we'll study by each way that if the primers that you have prepared are fine or not. So there are several methods to test that also, that whether the primers that we have prepared is fine or not. Dimer formation to nahi ho raha hai kahin pe, right? So we'll do that in next practical. We'll try to do that, right? So that's all for today's lecture. I guess this will be your third practical that you'll write in your plants. But then again, there's a slight twist here that I'll post it in a chat box that you will write a DNA extraction procedure for a different plants samples. So आपको ये एनालाइज करना है कि वो प्लांट सैंपल्स में पॉलीफिनोलिक्स ज्यादा है तो हमें कंपोनेंट कौन सा डालना है और हमें नहीं डालना है. सो बेसिकली आई ट्राई टू गिव अ डिफरेंट प्लांट सैंपल फॉर अ पर्टिकुलर ग्रुप, क्योंकि सबको डिफरेंट देना पॉसिबल नहीं होगा. सो आई ट्राई टू गिव फॉर वन ग्रुप अ डिफरेंट सैंपल, बट स्टिल यू विल डू इट सेपरेटली. सो this will exercise your mind, and you look for different protocols on the website, and you'll look for maybe different papers. From there you'll extract your protocols, and then at the end you'll give the reason also that why this protocol is being good for the isolation of the DNA from or RNA from this particular plant, right? So this experiment by evening I'll post, and by next week you can submit this experiment, right? How many of you have already submitted your practical files? I can see मुझे सिर्फ एक नजर आया था सुबह. किसका था वो? कौन था, किसी ने बहुत अच्छे से बना के सबमिट किया था, बड़े डायग्राम्स भी बनाए थे। किसने सबमिट किया बताओ नाम, तब तो मुझे पता चलेगा। There was some girl. There was some girl. Let me, okay. Ayushi, I guess. Let me open her practical. हाँ, आयुषी ने सबमिट किया था एंड इन अ वेरी गुड वे शी हैज, उसने बहुत अच्छे से, आयुषी हैव मेड द डायग्राम्स आल्सो एंड एवरी मैकेनिज्म, सो इट वाज डन इन अ वेरी गुड वे। सुबह से मुझे आयुषी का नजर आ रहा था। आधीश अथोयबा अभी भी टर्न्ड इन नहीं है, सैम भी नहीं है, एंड हु इज दिस? युवराज। युवराज, सैम मुझे आपका, अच्छा हाँ सैम का हो गया, योर टर्न इन राइट। आई आईटी सपोर्ट तो नहीं, जानवी नहीं है, मानसवनी, मानसवनी आपने मुझे मेल किया बेटा, आप उसमें अपलोड करो ठीक है।
Yes, ma'am. Actually, the one I made you, I made a mistake in that one, so I'm uploading a new one now.
Yeah, upload. So what you'll do in I'll not close this meeting, right? This meeting will go on, and what those who have not submitted, you all will stay here, and those who have not submitted, they submit by the end of this meeting this whole protocol, or those who have submitted, they can write their. Give me 5 minutes. I'll post the plants in the assignment section that which group will do which, so you can do that practical. So those who have done, they'll do that practical and they can submit, and others will do the previous practicals and submit. So by the end of this course or this class, if I don't see any practical files, so that student is not getting any attendance, so to remind it. Start doing it quickly, start doing it. I'm here only, जब हो जाए तो मुझे बताओ कि कर लिया है आपने. In the meanwhile, I'm preparing the assignment for the next practical. आप 5 minutes का ब्रेक ले सकते हो. I'm here, then you can join back if you all are tired and if you feel it's continuation. No problem. You can take 5 minutes break or a 10 minutes break. Fine. I'm still here and then join back. Hello.
Ma'am, am I audible?
देवांशी बहुत स्लो आवाज है. हाँ बोलो.
Ma'am, I can't able to upload. Can I send you on mail or on?
क्यों बेटा? एक्चुअली अपलोड आप मुझे मेल में भेजोगे तो मुझे अलग से फोल्डर बनाना पड़ेगा. जब यहाँ पे भेजोगे ना तो मेरे को सबका साथ में मिल जाएगा बेटा.
Ma'am, upload नहीं हो रहा.
क्यों नहीं हो रहा? होना चाहिए. क्यों नहीं हो रहा? आप ऐसा करो दिवांशी, फाइल सेक्शन में अपलोड कर दो. अपना नाम डाल के फाइल में जहाँ पोस्ट होता है ना वहां अपलोड करो. आई चेक.
Okay, ma'am.
ठीक है, डू इट, मेरे को बताओ फिर.
Okay, ma'am.
I have uploaded the practical and the names and the plants which students will do in the assignment section. Please check. Students, even still, I see some of the students, they have not submitted their practical files. And let me just name them. Adhish, Thoiba, Ishita, Kshitij, Nandini, Sahiba, Shakti has viewed but not turned in. Vivek has viewed but not turned in. Yuvraj has viewed. So please make sure that you submit this assignment today itself, and I will be marking attendance after the lunch only. So by that time it should be submitted, right? So in the meanwhile, all of you please switch on your camera for the attendance. Excuse me. Adesh Mishra. Ayushi Jain.
Present, ma'am.
Sam.
Present, ma'am.
Devanshi. Yes, Devanshi, are you there? Ishita.
Present, ma'am.
Janvi Singh.
Present, ma'am. Am I audible?
Yes, now you're audible. There's a lot of network issue at your end, Janvi. Kshitij.
Present,
Kshitij
ma'am.
Singh. Kshitij, are you there? Manaswani is there. Yes, Manaswani Shridhar. Namrita Pal. Yeah. Nandini Raj.
Present,
Present, ma'am.
Nehal Joshi.
Present, ma'am.
Rupal Joshi.
present, ma'am.
Sahiba. Sanjana. Shakti.
Present, ma'am.
Shivani.
Present, ma'am.
Am I
Tanvi.
audible? Sahiba,
Yes,
present.
Sahiba, you're audible. Ridiv.
Ma'am, he is
Vivek.
having some issues. Present, ma'am.
Vivek and Yukta.
Present, ma'am.
Yes. So all of you, please make sure that you submit your practical files so that you get attendance for today. Otherwise, I will be marking you absent, right? So make sure you submit your attendance, and mentor-mentee session. Those who are my mentees, I will be taking maybe after 2:30, after your ethics class is over. So Devanshi, just remind me once if I forget, right? Okay. So that's all for today. I'll see you tomorrow morning for the theory class. Thank you so much.
Thank you, ma'am.
Ma'am.
Thank you.
Ma'am,
Yes.
ma'am, I'm Kedav. I have submitted my practical and I'm present also.
Okay, Fine.